Without further ado, let's get started. Our first speaker today is Ken Pugh. Ken Pugh is President, Director of Research, and a Senior Scientist at Haskins Laboratory in New Haven, Connecticut. And he's Professor in the Department of Psychology at the University of Connecticut. He is also Associate Professor in the Department of Linguistics at Yale University, an Associate Professor in the Department of Diagnostic Radiology at Yale School of Medicine, and Director of the Yale Reading Center. Dr. Pugh is a cognitive neuroscientist, experimental psychologist who is best known for his work on the neural, biological, and cognitive underpinnings of research, of reading, and other cognitive activities. And in 2017, Dr. Pugh received the very prestigious Merit Award for extended research endeavors from the National Institutes of Health, and we are thrilled to have him here today. Welcome.
Thank you so much. It's really a great pleasure to be here today, and I just want to begin by acknowledging the long friendship and long history of shared work with Will Baker. Will is a force of nature in the field, in bringing science and practice together in venues like this and in many others. And whenever Will calls, I'm always here and always grateful to be part of this. So Will, it's great to be with you. I also want to begin by acknowledging, as Will did, that the work on the neurobiology of dyslexia or of reading disability, using another term, of language-based learning disabilities, using yet another term, the focus on brain basis owes much to the seminal work done by Norm Geschwind and Al Galiberter here at Harvard, very close to this place, focusing on the neural basis, that dyslexia can be understood as having a brain basis. Everything is an interaction between genes, brain, and environment, of course. But there's a clear neurologic basis. And for people like me who use modern neuroimaging tools, and you're going to get inundated with them today in my presentation, we owe a lot to the work that was done here and set the foundation on where to look in the brain, what to look for, and really how to understand these complex, bright, and fully capable children with specific learning disabilities. So it's a pleasure to be here. I'm here representing Haskins Laboratories, and I would just give a little shout out to, especially for those of you who are speech and language people, you're our people. Haskins Laboratories is a very historic place. We've been around since 1937. I'm the 5th president in 80-something years, whatever it is. We used to be in New York. We've been formally on the Yale campus and affiliated with both Yale and UConn since 1970. But it's a place where we focus on the biology of human communication in all its forms. So we focus on the mechanisms of speech perception and production and how they relate to reading, which is going to be, I'm going to torture you with how written language depends upon, is, as one of our linguists once said, parasitic upon good oral language skills. Shout out to all the speech and language people here. And we've also been, for many years, a place that focuses on what is universal in reading. You'll see a lot of this kind of work on reading and its psycholinguistics by my friend Kate Nation, who's presenting later. But when we look across languages, what is universal in successful reading and in the struggles of children with learning disabilities, and what's idiosyncratic to one or another? And I'll show you some of our data on English versus Spanish versus Hebrew versus Chinese. A little preview. We see a lot more similarity than difference when we look across languages, but maybe that's just because of what we look for. But we are very interested, and there, for those of you who've studied a bit on reading, you may know that some writing systems, Spanish, Hindi, especially Finnish, where we do work, are very transparent. And what that means is, essentially, if you know how it looks, you know how it sounds. English, not so much. We have words like pint that should be pint. And as linguists will say, the English writing system preserves morphology at the expense of phonology. So it becomes a more complex process to learn to decode efficiently and instantaneously. But is it really different than Spanish? And Chinese, which has a non-alphabetic system, where we do a lot of work on typical and atypical reading, is it really different? So we care a lot about what's universal and what's not. And Haskins Laboratories, he said with a certain amount of pride, not in my work, but my mentors, is the place where phonologic awareness was really first put forward and tested by Alvin Liberman, Isabel Liberman, and Donald Shankweiler, my mentors. And their next generation, Susan Brady and Hollis Scarborough and others. They asked a question at the beginning. If you want to figure out which kids are going to find reading easy or hard in third grade, what predicts it early on? And they found, and they tested the notion that, well, since reading is nominally a visual event, you have to use your eyes, and a lot of the currency at that time, in the 60s, was, well, if it's a visual event, it must be a visual problem. The work at Haskins was very important in establishing, and was, of course, we are very fortunate to have Louisa Moats here today, whose work expands on all of this. It's nice to see the mix of histories that will be presented to you today. But the importance of phonologic awareness. I say to you, say the word cat without the first sound, and you say at. Okay. And so this basic meta-linguistic, metacognitive understanding that spoken words are made of parts, I'm allowed to go this far, I was told. Okay, but no further. This far, but no further. Okay. I checked. What was I saying? Phonologic awareness is muy importante. And finally, the brain. We'll talk a lot today about what neuroscience can tell us and what it can't. Now, here's my remark to all of you. Most of you are not neuroscience background. You see a lot of stuff these days, brain-based this and brain-based that. People that do what I do get a lot of attention, often not deserved, for being able to show pretty pictures of the brain. You, as educators, are the people who are on the front line working with kids and shaping their brains. And if neuroscience has some value added to what we already know about reading from good psychology and good education psychology, then it damn well better show it. Because it's expensive, it's intensive. And so the question becomes, what is the value added of mapping into the wetware, just a jargony kind of a term, for psychologists, for educators, for reading specialists? There is a lot of talk these days about a term called educational neuroscience, where education and neuroscience are talking to one another. And we are very fortunate in our lab. I'm involved in 2 large in-school projects where we're collecting brain data and behavioral data in the AIM Academy. We've been there for, this was our first partnership. We've been there for over a year. And now more recently, the Windward School in New York. And we are educators and neuroscientists or cognitive neuroscientists working together to try to learn from one another and to explore the neurobiology of successful and challenged reading and language. It is cool to be able to see the brain, but what do we have that is value added? I will tell you right off the bat, that's what this talk is about. And I'm going to inundate you with brain terms, brain pictures, and I guarantee you there's no quiz in the morning. So you people do not need to remember the angular gyrus sits right behind the supramarginal gyrus, which sits above the superior temporal sulcus. But I'm going to be showing you this, and I insist that you pay attention and that you follow me into the brain. But I'm here to do something, I hope, if I do my job, much bigger than just giving you a lot of brain pictures that you will forget. I want to try to explain what we know, what we don't know. This is as important as what we know because there's a lot of hooey out there on brain-based this and brain-based that. And what the promise of educational neuroscience is, in my view, and what its limitations will be. But ultimately, follow with me what I'm going to share about what we know about the language systems in the brain and the learning circuits, reading. I'm going to be addressing 3 very big and chunky questions throughout this period. The first, and they're pretty obvious. 1, how does the brain reorganize itself and build circuits, which are just sets of connections among brain hubs, in order to become a fluent reader? And by the way, if you're a skilled reader in any language, and I use some clever tools like EEG, the estimate of how long it should take you to go from I to meaning. Cat shows up on the screen, and you begin to get the meaning of cat, is on the order of under 300 milliseconds. And 1,000 milliseconds is 1 second. So if you're a skilled reader of English or any language, you can get from I to meaning bloody fast, as the Brits would say, I think. And if you are not able to decode effortlessly and automatically and instantaneously, and each word takes your entire soul to pull off the page, then by the time you get to the end of the sentence, you kind of forget the beginning, and there's all kinds of bottleneck or limitations on comprehension and syntactic processing and morphosyntactic processing, et cetera. We all know this, and of course we know that kids who present with dyslexia, with specific reading disability, find this process of pulling the word off the page inordinately difficult. They are slow, labored, and in English, of course, error-prone. In languages like Finnish, Kate, I think you probably would agree, they are equally slow and labored and non-automatic, and this I'm going to show you why that happens in the brain. But they may not make as many errors in a language that has a transparent writing system. So if you use the Woodcock-Johnson accuracy test, you might not detect it as much in Finnish or in Spanish as you would in English. But really, as you'll see from my data, as I present this brain story to you, you'll see that that we really see a great deal of universality in all of this. But again, back to this issue. Skilled reading involves somehow reorganizing the brain and creating these fast connections. And the first question is, how does the brain do that? Uno. The second question I'll be addressing is, how does that go awry in some way in dyslexia? How do those bright, beautiful kids differ in the way these circuits are forming and the underlying structure and function and neurocognition that makes these circuits differ? So I'll be addressing the question of what does skilled reading look like and what does dyslexia look like? When I show you data on all of that, and that's 25 years of hard work for me and lots of other people around the world, I won't have answered why the dyslexic brain is different. So when people show you pretty brain pictures and say, we can tell you why dyslexia happens because it's here in the brain, then just give them a little smack upside the head. Because knowing how the brain is different is not the same as knowing why. And in order to know why, I'll show this as we go through my talk, we need a deeper dive into gene-brain mechanism. And that's what Al Galaberta and Norm Geschwind and their colleagues and our lab and others do. But when I get through all my stuff, and I'm telling you, here's what the typically developing brain looks like, and here's what the dyslexic brain looks like in terms of circuits and chemistry and connections, stuff that you need to know about if you're going to be skeptical, critical consumers, able to discriminate what's good science and what's not. But when I get to that point where I say, this is how the brain looks different, there's an exciting thing that we can do with that information, even though it doesn't explain. To say, well, this part of the brain becomes very well organized in typicals, and it does, right down here. And atypically developing kids, they tend to read with altered circuits. I'll show you that. Knowing that is not an explanation, but knowing it is in many ways a target for treatment. And I'll show you work from our lab and others where we address the third major question that I will take on, which is, given that there are differences in the brain, can effective evidence-based treatment moderate, modulate, minimize those brain differences? And that'll be the last part of the talk. In many ways, the $64,000 question, right? Okay, the brain's different. Can we, with the right early environment, see some normalization? When treatment works, and it works for many kids, but not all, we need to know whether that reflects the brain normalizing circuits or whether it's compensation. But more to the point, even the best treatment, and we've been lucky over the years to work with some of the best, like Benita Blackman and Maureen Lovett, the best treatment will not work for everybody. And so one of the things that we're currently doing, it'll be at the end of my talk, is using neuroscience, which gives us, and here's another buzzword for you, it's a Ken buzzword, exquisite sensitivity to individual differences. And I'll show you what that means. But by knowing something about those individual differences in the brain, we are exploring something very exciting right now, and I'll show you our latest data. And that is, can we use the exquisite sensitivity to individual differences thing to understand why treatment works for some and not others? And in understanding that, can we use knowledge of the brain to modify the treatment? And to put it in a colloquial term, can we use neuroscience to ask what works, for whom, and why? And I submit that, I used to give this talk, Will, in the old days. I'd come up here and I'd say, we should be ashamed of ourselves. We have so far to go, and we still have a long way to go. But I'm now seeing evidence that we actually can begin to deliver on this one, that we can use neuroscience to understand the complexity. All of you who work with kids who present with dyslexia know this well. They are complicated, and this is a heterogeneous condition. There are a lot of ways to get to the same end state, slow-labored and error-prone decoding. And the neuroscience gives us a real target, a real ability to get at those individual differences. So that's where I want to go. And let me begin with some overviews, some overarching ideas. These are also very much in line with the thinking in a laboratory like Haskins that is focused on the biologic specialization for speaking and listening. So I begin with the obvious. Babies don't need to go to school to learn to perceive phonemes. But they, of course, need what? Lots of talking to, lots of stimulation, lots of vocabulary, lots of sensitivity to rhyme and other things in birth to 5. But they come into the world wired. There's a great deal of brain specialization, especially along left perisylvian areas, and you'll see some of those, for talking and listening. And for those of you who are speech and language pathologists, you know that not all, but most kids develop oral language stereotypically and without going to school to learn to perceive and produce speech. And I say that in a silly way, but I want to emphasize why I'm saying it that way. I'm saying it that way because somehow that means at a neurobiologic level that the brain carries a great deal of biologic specialization. We are wired to talk and listen. And we are, of course, as Marianne Wolf likes to say, most certainly not wired to read and write. Written language has only been around for 5,000 years or so. The brain is not a reading machine by nature. It is, however, a talking and listening machine for damn sure. And so maybe the job of the reader and the process of reading is to reorganize systems that have evolved to do lots of jobs: language, vision, associative learning, attention, self-regulation, executive function, and turn them into something new. The new is what? A circuit that is allowing you to get from I to meaning bloody fast. And so how does the brain do this will be the focus of my remarks. But there can't be, in some sense, a brain specialization for reading. So ultimately, we are looking at what Stan DeHaan, who's a very influential figure in the reading and neuroscience, says, should be referred to as neuronal recycling. And this is the process whereby the brain takes populations of neurons that have evolved to do lots of jobs and turn them into a reading circuit. But this will not have the feeling, the flavor, the taste of oral language, which has so much biologic hardware, so much specialization, and on which we need to build reading, I would argue, from our studies. So let's first consider at a cognitive level what reading involves. As I said, you need to be able to get from I to meaning fast. And what that means in this division of labor diagram, this is based on Mark Seidenberg's division of labor model. Mark works with us on a lot of our psycholinguistics and our reading. Of course, many of you now know him through his amazing book, I love it, Language at the Speed of Sight, which really talks about how research and education needs to partner respectfully in venues like this. Anyway, if you are a child entering the literacy environment and you don't know anything about reading, you do know a lot of words. Many thousands of words that if you've been in an adequate pre-school, stimulative, well-resourced linguistic environment. And so you have a lot of information about two kinds of features of words: phonology, or the form, the sound, and semantics, or the meaning. So that is already in place, and it hopefully is in place really well, because the best predictors of reading from preschool are these oral language and vocabulary and sort of strength of representation on the right side of that. And of course, you know nothing about orthography when you begin. So that lower left circle is blank and needs to be filled in by who? By you and me and others who teach reading. And as you know, children who present with good reading will get from I to meaning fast. So you can imagine the I's out there somewhere, and there's meaning. And they pass through that division of labor fast. All those connections, which live where? In the brain. Those connections, the orthography, phonology, and sem, those are all well-structured and instantiated if all goes well. But the process of learning to read is building the base of the triangle. And when we get to the dyslexia part, I will remind you that one of the strongest presentations of developmental dyslexia is difficulties in creating automaticity at the level of grapheme-phoneme mapping, or orthography to phonology mapping. There's more to it, and you'll see the more to it in other presentations today. But this is a basic element. So how does the brain do that? So let's begin. Now I'm going to be doing something that I hate, which is I'm going to be giving you a rather oversimplified view of oral language in the brain. Like everything, let me tell you what the brain is really like when we map it. The brain is a nonlinear, self-organizing, dynamic statistical learning machine. And everything interacts with everything else. So how many of you have heard left brain people, right brain people? Of course, that's nonsense. That's a myth. But the left hemisphere is like muy importante for language. And if you're 7 years old or so, even less, I guess, and I damage the left perisylvian regions, these regions along this. Oh, I need to use the, sorry, we're being recorded, so, whoa, what happened? So I'm going to use this. Can you see that little pointy thingy? Well, then I may have to, yeah, they wanted us to use the other one because it's being broadcast. They're not seeing it. If you don't mind, let me use this one. If you damage the left perisylvian regions, this is the sylvian fissure, this is the temporal lobe, the parietal lobe, the frontal lobe, the occipital lobe, the cerebellum. If you damage this left distributed system, you are much more likely to lose language than if you damage the homologue in the right hemisphere. An interesting factlet for those of you who like neuroscience. Liz Bates, 30 years ago, showed beautiful data that demonstrates how plastic babies are. And by plastic, I mean good plastic, not like the kind you don't want in the Charles River plastic. If a baby is born with congenital lesion to the left or the right, they still develop language normally because that baby's brain is so bloody plastic and capable of reorganizing. But if you've lived in a world for a few years and absorbed language, the brain will tend to create special function in the left hemisphere. Now, in reality, when we look at the brain listening, this is about speech, about natural language. And when we look at the brain listening or talking, it's, of course, not limited just to the left. And we see very important, for those of you who are more in the neuroscience, more in the speech science, we see evidence for prosody and other slower unfolding temporal information in the right hemisphere, but the left hemisphere becomes dominant for language in most neurologically normal individuals. And so if I just want to give you a further sense of two kind of important subdivisions within this division in the brain. Remember, I'm trying to give you, I'm going to be telling you how the brain builds the orth-fon-sem division of labor and gets it so well structured in the brain that you can get from eye to meaning bloody fast. And so it involves a lot of these pathways. In the inferior frontal gyrus, a region that's come to be called Broca's area, and again, this is oversimplified for those of you who want to dive deeper into aphasiology and neuropsychology. But it still holds to an approximation, and it's reasonable. If I damage this motor area, this premotor area here, you are more likely to struggle with expressive language, and you may not struggle as much with receptive language. Okay? So this is expressive aphasia. So there's something about this tissue, these populations of neurons, that's involved in output, among 1,000 other things. This is your first exposure. I live in this world. All these systems are multifunctional and very highly interconnected. But nonetheless, this is an important thing for you to get right now, that there are areas in the left inferior frontal gyrus that are very important in producing oral language. And in the same way, an area in the superior temporal gyrus, this is the temporal lobe, referred to as Wernicke's area, if you damage this, there's a stroke or a bullet or some accident, then you are more likely to have receptive language problems and may not have the same degree of expressive. So again, this is an oversimplified dichotomy, but all of these systems here in left perisylvian cortex are supercritical for our amazing ability that we didn't need to go to school to learn to be able to produce and perceive speech and to entrain to somebody. For example, if Ken and I are talking, recent brain imaging from our colleagues at Haskins, Joy Hirsch, using a kind of you'll see these imaging things, that measures brain activity will show that when Ken and I are communicating, his Broca's area and my Wernicke's become correlated. The signal, the brain signals, become, it's called entrainment or hyperscanning. And it's kind of the cutting edge of the field. But ultimately, we are wired to talk and listen, and we ain't wired to read. Now, reading Reading, of course, involves a visual target. And as you all know, when you look at areas of the brain that are visual, it begins with the primary visual and primary and secondary visual cortical areas in the occipital lobe, the back of the head. That is where we see, as you know. So ultimately, in order to build that triangle that needs to be so well structured and feed forward, feed back, top down, bottom up, nonlinear, interactive, something, good stuff. You need to create connections that allow the visual signal that comes into the visual cortex here to get into the language machinery really, really fast. So there are a number of brain regions that are important and will play a role in the story that I'm sharing today. For example, in the inferior frontal, in the inferior parietal lobe, a region that was really discovered here by and talked about quite a bit by Norm Geschwind, the Wernicke's area, the angular gyrus, is a region that, if damaged, right here, will produce acquired dyslexia. So you will no longer be reading often if this region is damaged. And there's another region toward the bottom of the brain, down here, at the occipitotemporal juncture, that in the fusiform cortex. And again, you're not being quizzed on, oh, there's another thing I need to tell you, this is important. My slides are available if you want to get into the weeds later. My papers that are written, some of our papers have been written for general audience. So don't worry about, if you really do want to go deeper than just to understand what the brain can tell us for education, the slides are available. So don't worry about all these terms. And I don't think you need all these terms if you just want to get the message of my talk today, which is what do we know about these three questions. But in the left inferior ventral temporal, occipitotemporal region, there's other tissue that, if damaged, will produce acquired dyslexia. So, what does reading do? It's the creation of a circuit that allows you to create an orth-von-sem binding and get from I to meaning fast. It involves some of those brain areas because ultimately you've got to map from vision into language efficiently. So we know something from the background. We use many kinds of functional imaging. We use something called functional magnetic resonance imaging. Oops, sorry. Here. That's the vast majority of the data I'm showing you comes from fMRI. And we are there while you're lying in a scanner. I ask you to do things like read a word or hear a word. You'll see that next. And when you're doing that, those areas of the brain that are active will change the oxygen content of blood, because after a population of neurons has been working in some piece of the brain, there's a perfusion of oxygenated hemoglobin, oxygenated blood, to those neurons. And we can localize this in a magnetic resonance imaging machine to within a millimeter. So it's quite precise. It tells us a lot about where things are in the brain. And for this first presentation, for those of you who are not familiar, the where questions are easiest to grasp. But there are other kinds of imaging. For example, EEG, and many of you have probably been exposed to this. You put a cap on, it measures electrical activity on the scalp. The EEG is exquisite at getting the timing of these brain processes, because when we use fMRI, we're looking at populations of neurons that are active. And then there's an oxygen shift. And so it takes a few seconds. It's not real time, but we can find where they are. But when they're doing their job, when populations of neurons are talking to one another, this is intuitive, right? Then they're doing it in real time, in milliseconds. The EEG is on the scalp, measures the electrical activity under the scalp, and it's not so good at finding where those sources are, but it's really good at giving you information about what they're doing in time. So it doesn't take a rocket scientist, for God's sake, to suggest that maybe we should combine EEG and fMRI and get the best of both, namely where and when. And that's exactly what we do these days in all our research. And so you'll see, if you begin tracking this literature, many kinds of brain imaging that give us what? Give us information on how the brain is rewiring itself to get from I to meaning fast. So this is your first fMRI study, and I demand, I insist, that you pay attention and wake up right now, okay? You've had all my background, all the stuff about reading's hard, and you need to do it right, and you need to build the triangle, and obviously it happens in the brain, not the elbow, and why is it important to know the brain? And we'll keep going back and back and back to that theme. But now you get your first taste of brain data. So this is an fMRI study. Hundreds, I think we're over 1,000 children who've gone through the scanner with us. Me and all my family and myself, more times than I can remember, go into a scanner and we localize where stuff's happening, okay? Now, in this experiment, which is going to serve as a model for the rest of the talk, or for at least most of the talk, there is a simple game that the subject, the person, is playing in the scanner. They are either hearing words like cat or door, and they do its job. They say, is it a living thing or is it an object? Now, I know you're all like really hip with this, so cat, living. The door thingy, not living, okay? So why am I doing that? Well, I'm forcing the participant to go from ear to meaning, and I'm mapping where that is. And in another condition, they get the same words, counterbalanced across subjects, this experimental design stuff, where they are making the same judgment, but the word comes to their eye. So this is looking at reading versus listening. This is the basic question. How does reading sit with listening? And what you're looking at in the colors, this is 20 adult subjects, and none of them struggle with reading, and they're basically skilled adult English readers in this map. What do the colors mean? You ready? Not hard. These are statistical analyses showing where activity is. And what activity? Activity somehow related to listening or reading. Now, in red, here, is a part of the brain which is called the primary and secondary auditory cortex. I'm giving you a heads up because I'm asking you this question. You. Which, if only one of the two modes, listening or hearing, activates the things in red, and it's the auditory cortex, which one is it? Hearing. You pass. Okay, now the next one. In the back of the head, you see this little yellow thingies? If I turn the brain around, you'd see it clearly. Which one does it respond to uniquely? Seeing words or, okay. So if I don't see that, I might as well quit the field and like go sell cars. Because this is the auditory cortex, and it better light up when you hear a word or when you hear a sound or a musical note. And in the same way, in the back of the head is the visual cortex. But what's really amazing, drum roll please, is all these areas like Wernicke's, remember I told you Wernicke's and Broca's and the angular gyrus, and they are all painted green. What the heck is green? You ready? This is the rest of the talk right here. These populations of neurons in skilled adult readers of English don't give a damn anymore whether the stimulus came to my ear or to my eye. Now, these are parts of the brain, most of them, that are integral part in an illiterate or a pre-literate brain, somebody who doesn't read. These are all parts of what system? Talking and listening. But in a literate brain, these areas are changing. So, learning to read is changing speech. Is that important? Is it intuitively obvious? Would it be obvious to your grandmother if your grandmother were Mrs. Orton or Isabel Liverman or any of our dear friends? No, yes, they would. That's not surprising. Good reading is somehow dependent on speech. But does this matter, and does this tell us something that teachers need to know and neuroscientists need to explore? Well, I'll tell you one thing that's kind of come up here, obviously. I'll jump ahead a little bit. if somehow building a good reading circuit, and if I'm about to show you that really matters and predicts and is important to intervention and everything else, then it kind of reinforces what good teaching and what Orton Gillingham and Wilson and others tell you. You must strengthen the oral language skills in order to allow reading to come and sit upon the machinery of talking and listening. So green matters. So I won't go into all these details, but I'm happy to share the papers. Those areas that I've just alluded to, there are 3 broad hubs. This is just to give you something to hang on to. To what question? Question number 1, how do skilled readers build circuits? First thing you need to know is that print and speech become, it's called amodal. They sort of become the same thing in the brain. And the second thing is that there's a lot of areas that take on very important roles. So in learning to read, what is learning to read according to Ken's stupid diagram? Building the orthography to connect to the phonology and the meaning. That's a good diagram for you to hold on to, because this is complicated stuff. Building that diagram depends in an emergent reader, beginning reader, first grade, who becomes accurate but not automatic. It depends on the integrity of a number of areas, including an extended parieto-temporal region, including the supramarginal, angular, and superior temporal gyrus. Remember, I told you there's no quiz on these terms, but I damn well will use them, okay? And you can read about them later. And then in the same way, in the inferior frontal gyrus, extending from Broca's, there's a lot of tissue that's very important. And in a beginning reader, the feed-forward and feedback connections between these 2 parts of the brain are very important in building the base of the triangle. And if the base of the triangle goes well, what happens to a reader as they go from first grade to third grade? Well, they go from being accurate to being fast, as Kate will tell you, okay? And becoming fast, neurologically, we've now found, in every language we've looked at, involves developing a fast mapping system, much along the lines of what you've talked about in the past, that in the occipitotemporal region, that allows that as kids get older and more automatic, faster and more fluid, there is a shift in energies toward this region, the occipitotemporal. And in fact, there's a lot of data from a lot of labs, including ours. Ours was among the early studies for this, but it's shown up in many languages, that as kids go from being accurate to automatic, there is the development of specialization in this part of the brain. Now, for those of you who care about neuroscience in any deep way, the population of neurons in these extrastriate and occipitotemporal regions are designed by nature for pattern recognition. In the right hemisphere, there's a homologue region that is designed and specializes for faces. But in the left, these fast mapping systems become entrained to print. And you'll see what that means. But at the end of the day, skilled reading, to the first question, what does the brain do? Well, it links print and speech, and it develops these fast mapping systems. And I told you why that's going to be important, because if it's different in dyslexia, and it is, we'll be there in a minute, then it also suggests that if we're looking at treatment, we should be looking and targeting whatever it is that computationally these regions do. We need to understand it so that we can target the treatment to that. For those of you who also care to go one step further, the brain is a really complicated machine, but the cerebral cortex is not where all the action is. The brain has both cortical and subcortical elements. One in particular is the basal ganglia and the thalamus. And these areas work with the cortical regions in order to create automaticity, learning, representation, etc. And so it's not surprising. We did a study of 100 children who were just emergent readers using fMRI, and we were able to, it was a very complicated study, but what we were able to do is identify those neural populations of neurons that kids who were closer to succeeding or those who were further away were using differently. And the system involves not just the areas that I was telling you about, sorry. the parietal and the ventral, but also the thalamus, the pulvinar nucleus in particular, and frontal areas that are involved in executive function and response inhibition and self-regulation. And you all know as teachers that kids will learn nothing if they're not able to self-regulate. And so the learning circuit is, of course, not just language, not just orth, phon, sem, not just cortex, but also subcortex. But we care a lot about how print depends on speech and how speech changes with print. So we are interested in the bidirectional relationships. So this is a study by one of our dear colleagues and collaborators, Manolo Carreras, where they looked at illiterate versus literate guerrilla fighters from Colombia, South America, in a scanner. And what they found is that a lot of those areas I've been talking to you about, particularly the parieto-temporal, are thicker in their gray matter volume. These are areas that are part of the speech communication system and the representation of knowledge, of vocabulary, et cetera. These areas are thicker in their gray matter density if you've learned to read. So learning to read is changing speech. This was a study done by Stan DeHaan, the famous DeHaan, who finds his way into my slides and my dreams once in a while. Actually, my nightmares more. Not those kind of dreams. Kate! I'm a grandfather, for God's sake. No, the other kinds of dreams, like, oh my God, he's going to beat me to be the first to publish something. DeHaan's great. But this is a beautiful little study where he took illiterate adults and he made them literate, taught them to read. And what you could see is that areas that are involved in speech, like that Wernicke's area, become more active for speech. So learning to read changes speech. We don't usually think about that. But if it changes speech and it's bidirectional, then in some way reading must be dependent on speech. So here we go. You ready? I'm going to go through this now fast. 1, we were doing a longitudinal study in my lab. We were tracking 100-something kids in New Haven. They came into this study. This one, they were a little bit older because at that time we didn't think we could do fMRI or have kids keep their head still in the scanner. It's you know, you have to keep your head still. So they were 7 and 6. So they were a little bit older. But when they came into the study, it was a full range of abilities, and a lot of the kids were presenting with high risk for dyslexia diagnosis. And we tracked them for 2 years. And when they first came in, we measured everything in the kitchen sink. Genetics, neurochemistry, and you'll see that briefly, structure of the brain, function of the brain, cognition, learning, speech perception, production, and everything. And then we watched them grow for 2 years. And we asked this question: what predicts reading in 3rd grade from gene, brain, cognitive measures at 1st grade? And we tested in this first study the kids at time 1. So we got 2 years to wait. So what do we do at time 1? Well, we said, let's ask whether the ability to say the word cat without the first sound, and you said at, whether that ability is somehow associated with a different brain organization for print and speech in 1st grade. What matters about phonologic awareness? And this is what we found. Now, this is the last bit of neurology I need you to pay attention to. You ready? If you are looking at these are axial slices. They're horizontal slices through the brain. Not slices, because otherwise we'd be all dead. They're camera slices. Right? It's as if you're looking this way. The left hemisphere is on the right side. That's the left hemisphere on the right. That's the right hemisphere on the left. That's all you need to know. The left hemisphere is like a little bit important for language, okay? As I said before. And what we're finding in Wernicke's area and in the secondary auditory cortex are populations of neurons in 7-year-old kids who vary, because we recruited them to be the full distribution of risk for reading disability, from high risk to average reading likely and to precocious. And the kids who were at high risk were having trouble saying the word cat without the first sound, because that's a primary biomarker, a cognitive marker. So when we separated the kids, actually the analyses were done continuously for the researchers here, but then this is shown with a median split. These are kids who are likely to be struggling with reading in the next 2 years, big time, because their phonological awareness scores are quite low. And these are kids who are not. And this is them before they really are doing a whole lot of reading. They're just starting. And what this interaction map shows is that if you look at the behavior of this part of the brain that is specialized for spoken words, to spoken words, in gray, it activates just fine for everybody, because it doesn't care whether you can say the word cat without the first sound and you said at, because it's built for speech. But look what happens for print. If you have a better metaphonological awareness, it's not reading. We've covaried out reading. We did all kinds of things. What this shows is that this speech area responds and activates to printed words at age 7 if you can say the word cat without the first, I'm being silly about that, but if you have phonologic deficits, you have not yet gone from the visual area over to the speech areas and started to change them. And does this matter, guys? Well, this was the payoff. 2 years later, we brought them back. And in a paper that my speech language pathologist postdoc, John Preston, who's now a professor at Syracuse and stays at Haskins to my great happiness, he's first author. And we took that same group of kids and we said, okay, now they're 2 years older. What was it when they were 7? You ready? I'm going to do this in a jargony way. What was it at age 7 that predicted reading in 3rd grade? Was it what the brain looks like when it reads words? Because we had measured it. Was it what the brain looks like when it listens to words? Because we measured it. Or, given the punchline you know I'm setting you up for, is it whether it uses the same tissue to do both jobs? And the answer was, using the same tissue at age 7 to do both jobs is predicting reading outcomes with an endless variety of regression controls, and the auto-regressor from the same measure for the researchers here, everything removed. Above and beyond that, covarying out for total activity and all kinds of things. The best predictor of 3rd grade reading. Oh. And And this is 3rd grade reading scores on the Woodcock-Johnson broad reading test. How many of you know the Woodcock-Johnson? This is your score in 3rd grade, okay? And if you're high, that's good. And if you're lower, that's not good, as you know. And this is whether or not you use the same tissue to do both jobs at age 7 in left perisylvian cortex from parietal, temporal, and inferior frontal. The best predictor of reading in 3rd grade was a brain that in 1st grade was already solving the puzzle of mushing, in a good way, mushing print and speech together. So you damn well want to be focused on interventions somehow at the beginning that are language hip and sensitive. And I'll skip that because we're getting low on time. I just want to mention this also. This is also a brief aside. Does this only tell us about, is this print-speech connection only about, the beginning of reading? So this is a paper we published with Don Shankweiler, and this was a group of young adults who were struggling with reading comprehension. This is an area that several of the speakers do a lot more work than I do. And we had them processing sentences, not single words, that were either subject or object relative sentences, which are syntactically harder or easier. And the sentences either came to, because we do this all the time, the ear or the eye. And what we found is that those brain areas that predicted syntactic processing, sentence processing, and didn't care on average whether you were reading or listening because you were a good reader. And what we found is in the inferior frontal gyrus, this is the left inferior frontal, we are looking at people whether they have more integration of print and speech for sentences and for comprehension or less. And this is a really kind of a striking finding. We have a correlation of 0.7, it's a big correlation between reading comprehension scores and whether you have the same populations of neurons doing syntax, or whether they're anatomically dissociated. So it kind of reinforces this story that I'm killing you with, okay? Is this true in other languages? Well, I wouldn't be asking the question if I didn't have an answer. It's just like when you go to court. Don't ask a question that you don't already know the answer to. I already know the answer, because we published this in PNAS with a group of crazy people from New Haven, Hebrew University, the Basque Center in Spain, and the National Central University in Taiwan, and we looked at skilled readers, and in this PNAS study, here's the take-home. When it comes to the tendency, even in a non-alphabetic writing system like Mandarin Chinese, for the good skilled readers to have this integration where populations of neurons no longer care, don't give a damn where it comes from, we found that to be invariant, and we couldn't find a jot of difference in Chinese, beautifully transparent, muy beautiful Spanish, crazy Hebrew, because unpointed Hebrew, if you're reading like the newspaper, there are no vowels in the unpointed Hebrew. And my colleague Ram Frost studies unpointed Hebrew. And then, of course, English with words like pint that should be pinned. So I'm going to have to skip this, but Kate, this was the slide that... so one of the things the speakers are going to be doing today is they're going to be talking, several of them, are going to be talking about how we get from I to meaning and what kinds of information are used, morphology, orthography, vocabulary, et cetera. And we are now, but actually, just quickly look. So that's the data. If you have more sensitivity, sorry, just 2 seconds, more sensitivity to consistency, you have a positive, and there it is. Okay, so for the rest of you people, I thought I'd have more time. So let's, I'm going to also do short shrift to something else here, which is, if all of this is true, and we really do see this as universal, and in some ways it reinforces what your grandmother knows, if your grandmother understands that good reading is parasitic on speech. But it took us a long time to see whether, is this really a biomarker of success? And the fact that it better predicts reading in older kids than any other measure was really striking. And also suggests value added in the brain, remember my original question? Because it gives us insight into why phonics and phonologic treatments might be important. All right, but because this suggests that the ability of the machinery of talking and listening to be able to transform itself to do what DeHaan calls neuronal recycling is really important in reading success, that would suggest that perhaps children at risk for reading difficulties are not just having problems with, say, the word cat and without the first sound, which is meta-phonological, but also with the quality of perception and production in preschool. And I can begin, because I can do this one quick, with a study we did looking at one of the known risk markers for reading problems, late talking. And in that study, we found that kids who were late talkers were on average still reading worse, and were reading worse. Not every late talker will be a poor reader, and not every kid with reading problems will be a late talker, so don't but there's a statistical relation. And it suggests, and we found even at age 7, that we could find anomalies in the basal ganglia and the thalamus and in Wernicke's area. And why I'm showing this slide is to remind you that good reading, mind myself to remind you, good reading starts early. In the sense, not that you teach babies to read, that's insane, but that you make babies ready to read by talking to them wonderfully well. And I don't have time to go into this, but I just want to highlight it, and I'm happy to talk about it with people offline. We also, and especially for the speech and language people, we also look at really interesting tasks, something called the modified auditory feedback, where we manipulate the signal that you get from your own articulation, and we play with these parameters. And it gives us a window on the feed-forward feedback integrity of Broca's and Wernicke's. And what we found, I won't have time to unpack this, but what we found was that in Dutch and English cohorts, that this low-level basic connection between the Wernicke's and Broca's talking to one another is disrupted in dyslexia. And dyslexia, of course, not defined on subtle speech mechanics, but we do see a disruption. We published these studies are all published, and I'm happy to share them. Okay, today, as many people are going to be talking about spelling. I just want to, there isn't a lot of research on spelling yet, but what we do know about spelling is that it uses the same overlapping brain networks in the occipitotemporal and the inferior frontal gyrus as reading and writing. So while good spelling is not just, oh, they're all the same and we can reduce one to the other, okay? It is the case that at a neurologic level, from what we know thus far, that spelling hinges on the integrity of the same system. And it's likely, therefore, that reinforcing spelling or reading should have some positive effect. Brain science hasn't told us much at all on this. Okay, let me just take 2 minutes to finish. As I mentioned, dyslexia looks different in the brain. And here's how it looks. Those left posterior regions that I talked about, this parietotemporal and the inferior frontal, show anomalous activity and a failure to create print-speech binding. This is a study by Eraldo Palasu, and in shallow languages like Italian, or irregular languages like English, or intermediate languages like French, what dyslexia looks like is a disruption of these left posterior regions, and that is present in all languages. And it's also the case that the disruption is not just in activity, but in what we are thinking of, and you'll remember this, connectivity, which is how those populations of neurons communicate with others. And so I'm out of time, so I'm trying to quickly answer the second question, which is, what does dyslexia look like in the brain? It looks like a failure to develop these systems and to create the binding. And with that in mind, if I may just show my last slide, these differences are well established. And we do a lot of work, and it's not for today, on how they arise from genes and neurochemistry. And it's a whole other talk, and I'd love to give it to you. But I want to end with these slides. Knowing that the brain is different in dyslexia is great, but it's hardly what we care about or you care about. What you care about and what parents care about is, can neuroscience give us insight into treatment? And so I will tell you that we already know from a lot of work, ours was among the early, the Shaywitz-Pugh 2004, we had subjects, children from Syracuse, spend 1 year in Benita Blackman's wonderful evidence-based treatment. And we scanned the brain pre, post, and 1 year follow-up. And guys, the good news, if the treatment works, we get a normalization of many of those left hemisphere regions, including, drum roll please, this important occipitotemporal region. So knowing how the brain differs, diving more deeply into mechanism, we begin to see, yes, well, when treatment works, we can see some degree of normalization if the kids are young and the treatment's right. But as Louisa will tell you, in any good treatment study, in ours it was 75% of the kids were responders. That means 25% are not responders. And I'll end with this. Knowing that treatment can have a normalizing effect, the power of neuroscience, I submit in my last moments before the hook comes, I submit to you that the power of neuroscience in treatment is not what treatment looks like when it works, because what it looks like when it works is the brain looks more normal. It's understanding why it works for some and not others. We are currently and this is my last. We are currently we are doing a study that we've just run 183 kids in Atlanta, Robin Morris, Maureen Lovett, for those of you in the field. And on average, the treatment was effective in those areas that I told you about. But this is not a question about on average. So what we're now looking at is, what is it in the brain? What can the brain tell me about why some kids move and some kids don't? And by knowing what is going on in the brain of kids who don't, and where the fragile places are in the brain, I can begin to tailor the treatment to the sources of those fragile connections. I'm not going to show you the rest of my slides. Thank you very much. Three.